Hello everyone. So we are going to start a new topic, that is the biosynthesis of purine and the pyrimidine nucleotides. So we all know that these purine and pyrimidine nucleotides, they are the integral part of the genetic material which are recognized or which are known as the DNA and the RNA in the human body. We all know that this DNA, they are generally the information carrier from the parents to the offspring, and we all know that the function of any cell or any organism or any organ, you can say, it is totally controlled by the amount of the information which is carried in that particular cell or in that particular organism. And this amount of the information which is carried, it is carried by the virtue of the nucleic material which is recognized as the DNA and the RNA. So let's start with a new era of study, that is the synthesis, or you can say the biosynthesis of the purine and pyrimidine nucleotides. So how these two nucleotides, they get synthesized in the human body. We are going to study their basic steps, how this synthesis takes place, right? So without wasting any time, let's start. So now if we see the background, you can see here I have shown you two basic structure of the, you can see here the structure of the pyrimidine and the structure of the purine, right? So now the purines are generally the adenine and the guanine. So these are the purines which are found in the human body, or you can say in any cell, right? Moreover, the pyrimidines are, or you can say the examples of pyrimidines are the uracil, the thymine, and the cytosine, right? Now you see, we know that this purine and pyrimidine, they are the name of the parent compound of two types of nitrogen containing heterocyclic aromatic compound, right? So this purine name, it is the parent name for the adenine and the guanine, whereas the pyrimidine, it is the parent name for the uracil, thymine, and the cytosine, right? Moreover, this adenine and the guanine, we know that they are the principal purines of both the DNA as well as the RNA. But what about the pyrimidines? The pyrimidines that occur in the DNA are generally the cytosine and the thymine, but the cytosine and the uracil are the pyrimidines in the RNA, because the cytosine, it is same for both, but the thymine, it replaced with the uracil. So if we write the genetic code, we used to write ATGC and AUGC, right? So for T, it is replaced with the U in the RNA, right? Now you see, we have already discussed regarding this part in the introduction of biochemistry, what I have taken in during the classes. So we know that the nucleoside, this is a structure which is basically formed by the combination of the nitrogen base, or you can say a nitrogenous base and a sugar. So when a nitrogenous base is combined with a sugar, it forms the nucleoside. But the nucleotide, it is the combination of the phosphoric acid group with that of the nucleoside. So earlier you will have the sugar. Into the sugar, if you are adding the nitrogenous base, you are getting the nucleoside, and to this nucleoside, if you are adding the phosphorus, then you are getting the nucleotides. Sorry. Now you see, there are two pathways for the synthesis of the nucleotides which happens in our body. The first pathway, it is the de novo synthesis. The next one, it is the salvage pathway, right? So the de novo synthesis pathway, it is the biochemical pathway actually, where the nucleotides, these are synthesized from new simple precursor molecules. So I have shown here, you can see, it is the synthesis of the purine. For purine, I am saying it is the synthesis of the purine based step by step on the ribose phosphate. We are starting with the initial sugar molecule, right? Whereas the next type of synthesis, it is the salvage pathway, where it is used to recover the bases. So after the destruction of the bases, and the nucleotides formed during the degradation of the DNA and RNA. So whatever the nucleotide or nucleotides or bases it forms after the degradation of the RNA and the DNA that can be again reused for their synthesis, and this reusing pathway it is known as the salvage pathway. So here what actually happens, there is a addition of the ribose 5-phosphate to the preformed purine bases, and addition of phosphate to the purine nucleoside. So this generally takes place in the plastid, whereas the new de novo synthesis is generally takes place in the liver and the cytosol, right? So now this is the overall chart for the synthesis of the nucleotide, biosynthesis of the nucleotide. If somebody asks you, you can describe in this particular way. See, it have been divided into two basic parts. First one it is the de novo synthesis, next one it is the salvage pathway. So in the salvage pathway you are going to get the purine as well as the pyrimidine. Again in the de novo synthesis here also you are going to get the purine and the pyrimidine. Now this purine is going to give you the first product, you can use the word pro because it is the initial product, right? It is the IMP. It stands for basically the ionosine monophosphate. So from this IMP you are going to get two component, one it is the AMP, another one it is the GMP. The AMP it stands for the adenosine monophosphate, GMP it stands for the guanosine monophosphate, whereas this ATP it forms from this AMP only, it is the adenosine triphosphate, whereas this is the guanosine triphosphate. Similarly from the UMP, from the pyrimidine, if you are getting the UMP it is nothing but it is the uridine monophosphate. So it get converted into the DUMP, which is also known as the deoxyuridine monophosphate. From here you are going to get the DTTP, it is the deoxythymine triphosphate, whereas this from this UMP you are going to get the UTP, it is nothing but the uridine triphosphate, and from here you are going to get the CTP. So it is the basically it is the cytosine triphosphate. So you are getting everything here. Actually you can see cytosine, uracil, thymine, everything here, as well as adenine and guanine, right? So this is the basic pathway actually how it takes place. So we are going step by step with the synthesis. So let's start the source of different atoms for the purine ring. So how this purine ring it is going to form. So we know that this is the purine ring. So here I have shown you in the figure, you can see here this particular part, this N1, this N comes from the aspartate. Again in this C2 here, this C2N as well as the C8. So generally these are contributed by the formate, right? Then the N3 and the N9 here, the N3 and the N9 it is coming from the amide group of the glutamine, as well as the C4, C5 and the N7 it comes from the glycine, and the C6 it comes from the bicarbonate. So this is how it forms. You can see this structure for the basic learning, right? Now let's start with the purine synthesis. So I'm going to give you the details with this synthesis, so you are going to have both the structure as well as the description for this. So just see step by step. So let's start with the basic step. So it is that we know that the ribose 5-phosphate of the carbohydrate metabolism, it is the starting material for the synthesis of the purine nucleotide, right? This is the first thing. So here I am showing you as a alpha D ribose 5-phosphate. It is the starting material, right? Now what is the step? So it is going to start with the ATP. So it is going to react with the ATP to form the phosphoribosyl pyrophosphate, PRPP. You remember this term because after this I am going to use only PRPP. The PRPP it is the phosphoribosyl pyrophosphate. Then afterwards you see here, once the PRPP it has formed, the next step it is nothing but it is the transfer. So here what will happen, the glutamine it will transfer its amine nitrogen to the PRPP to replace the pyrophosphate and produce 5-phosphoribosylamine. So here you are getting the phosphoribosylamine, right? And it is catalyzed by this reaction, it is catalyzed by the PRPP glutamine aminotransferase. So this is the reaction, this is the first step. So this reaction it is a committed reaction. You should know that this is a committed reaction because once it will start it is going to get end, right? The next step it is, you see here, now here we have already got the phosphoribosylamine. So now this phosphoribosylamine it is going to get react with the glycine to glycine in the presence of the ATP. You can see here, see the glycine is there in the presence of the ATP, right, to form the glycinamide ribosyl 5-phosphate, or you can say glycinamide ribotide, which is also regarded, or which is also said as a GAR, and this reaction it is catalyzed by the GAR synthase. Whereas once it has formed, now what will happen? The next step, it is the N10-formyl tetrahydrofolate. It's a molecule, N10-formyl tetrahydrofolate. It is going to donate its formyl group, and the product from is known as the formyl glycinamide ribosyl 5-phosphate, and this is catalyzed by, this is FGAR, it is catalyzed by the GAR formyl transferase, right? The next step, it is, now you have already got the FGAR. Now what will happen? You see here the next step, once the FGAR has formed, now the glutamine, it transfers the second amino group to produce the formyl glycinamide ribosyl 5-phosphate. This is the FGAM. Earlier it was FGAR, so now this glutamine is going to transfer its second amino group to give you the FGAM, right? Moreover, once this FGAM has formed, the next step will be simply the imidazole ring of the purine. It is now going to get close. So it is going to get close. This ring, it is going to get close in a ATP dependent reaction to yield. It will yield 5-aminoimidazole ribonucleotide. It is here I have shown you 5-aminoimidazole ribonucleotide, and it is catalyzed by the general AIR synthase enzyme. The next step, it is here, what will happen? We have already studied regarding this thing. So now the next step, it is the incorporation of the carbon dioxide, that is the carboxylation will take place, right? The incorporation of the carbon dioxide will occur to yield the aminoimidazole carboxylate ribosyl 5-phosphate. So it is catalyzed by the enzyme carboxylase. But you should remember that this step, it don't require vitamin biotin or ATP as a cofactor for this, right? So the next step now, it is that the aspartate. Now it is going, this aspartate, it is going to get condensed here. You just see, while I am explaining, you just see in this structure, right? Now this aspartate, it is going to condense with the aminoimidazole carboxylate ribosyl 5-phosphate to form aminoimidazole 4-succinyl carboxamide ribosyl 5-phosphate, and it is catalyzed by the enzyme synthesis. So it is the product we are getting right now. So carboxyl, here it is the product. So it is the carboxy aminoimidazole ribonucleotide. The next step, it is nothing but the step 9. It is the adenosaccinylase. It will cleave off the fumarate from this structure. The adenylosuccinate lyase, this enzyme, this adenylosuccinylase, actually it is going to cleave off the fumarate. It is going to remove the fumarate, and only the amino group of the aspartate is retained to yield the structure. What your structure you are going to get? You are going to get the 4-carboxamide ribosyl 5-phosphate. Right now, the next component it is going to get here. It is again the N10-formyl tetrahydrofolate. It will come. So N10-formyl tetrahydrofolate, it will donate 1 carbon moiety to produce 5-formimidazole 5-carboxamide ribosyl 5-phosphate. So this reaction, it is catalyzed by the formyl transfer. So we are coming to an end. The next step, it is simply nothing but, you see here the final reaction, it will be catalyzed by the enzyme cyclohydrolase, which leads to the ring closure. Now the ring is going to close. It will lead to the ring closure, and the product obtained, it is nothing but, it is the inosine monophosphate, which is also abbreviated as IMP. It is the parent purine nucleotide from which other purine nucleotides can be easily synthesized. So IMP, it is generally considered as a parent product, right? So here if we see, so how from the IMP this AMP and GMP will form. What are the steps? The AMP, it will finally give the adenine and GMP will give the guanine. So basically you see synthesis of the AMP. So this parent product, that is the IMP, it is the immediate precursor for the formation of the AMP and GMP. You should know that. It is the immediate precursor, means from IMP only GMP and AMP it is going to form. Now you see the aspartate, actually what will happen, the aspartate, it condenses with the IMP in the presence of the GTP to produce the adenyl succinate, which on cleavage from AMP. So you have got finally the adenine, right? Now synthesis of the GMP. So how GMP it is going to get synthesized? Simply the IMP it will undergo one NAD dependent dehydrogenation to form the xanthosine monophosphate, which is also abbreviated as XMP, right? Now the glutamine, the next step it is the glutamine then transfer its amide nitrogen to the xanthosine monophosphate XMP to produce the GMP. So initially it will form the XMP, then the glutamine will convert the XMP into the GMP, right? Now you should know that the 6-mercaptopurine, it is an inhibitor for the synthesis of the AMP as well as GMP. So 6-mercaptopurine, it can be used in the treatment of cancer because they can be directly used for inhibition of the synthesis of the AMP and GMP, which will ultimately interfere in the process or synthesis of the nucleotide material in the cell, right? Now it acts as an enzyme. It's basically this mercaptopurine, it acts on the enzyme adenyl succinate of the AMP pathway and the IMP dehydrogenase in the GMP pathway. So where it acts, sometimes if somebody asks you where 6-mercaptopurine acts, you can say it acts in the adenyl succinase of the AMP pathway and IMP dehydrogenase of the GMP pathway. So here I am again showing you how it starts. So let's start with the IMP. So here aspartate molecule will come. You are going to utilize one GTP molecule to finally you are going to get the adenyl, this particular component, that is the adenosyl succinate, right? From this adenosyl succinate you are going to get the AMP, right? Similarly here it depends on a NAD dependent redox process. So here you are going to get first the xanthosine monophosphate. Again the glutamine will enter here along with the ATP molecule to form the GMP. So this is the structure, basic structure, how this AMP and GMP forms from the IMP. Clear? Now let's start the salvage pathway. So what actually happens in the salvage pathway? Nothing. It refers to the formation of the purine nucleotides by how you are going to form. Number one, addition of the ribose phosphate from the Prpp. We have already discussed this Prpp. So addition of the ribose phosphate, it will take place. So it will come from the Prpp to the preformed purine bases, which has come from the degradation of the DNA and the RNA, right? The next step, it involves the addition of the phosphate to the preformed purine nucleotides. Phosphate will be added. Now once the phosphate will be added, it is going to get the nucleotides. From the nucleotides, you know how it gets synthesized. Now, what is the significance of the salvage pathway? So, basically, you see, salvage pathway provides a pathway for the utilization of the purine bases, which derive from the diet. Whatever you take in your diet, if it have a something means like a component like purine bases that can be easily utilized in the body, and the normal turnover of the nucleic acids in the human body, right? Moreover, in the erythrocytes, we know that in the erythrocytes they are devoid of the nucleus. So once the nucleus it is not there, definitely there is not the nucleus. It is not. If the nucleus is not there, definitely there is no chances of presence for the DNA and the RNA. But in the erythrocyte, what actually happens? In the erythrocyte, the de novo synthesis of purine nucleotides does not occur. Why? Because there is an absence of the enzyme Prpp amidotransferase. And the requirement of the purine nucleotide is met by the salvage pathway. Clear? So let's start with the synthesis of the pyrimidine. So again, the de novo synthesis, it takes place in the liver, cytosol and the mitochondria, whereas the salvage pathway is always takes place in the plastic. So de novo synthesis, it starts with the synthesis of the pyrimidine nucleotide refers for the formation of the pyrimidine ring structure followed by addition of the ribose phosphate step by step, whereas here formation of the pyrimidine nucleotides from the pyrimidine bases. So I am showing you the structure of the pyrimidine. So here it's the structure, you can see here it all comes from the aspartate and remaining this come from the glutamine and this come from the bicarbonate. So here you can see these are the sources of different atoms for the pyrimidine ring. Now let's start the synthesis of the pyrimidine ring. Let's explain. It will get completed in a single slide. So initial step, so two molecules of ATP, it is going to combine with a bicarbonate and the glutamate in the presence of the water, and this reaction it will be forwarded or catalyzed by the enzyme carbamyl phosphate synthase 2. It will lead to the formation of carbamyl phosphate, right? Once you are going to get the carbamyl phosphate, the next step is the addition of the aspartate. So the addition of the aspartate, an enzyme will come, that is the aspartate transcarbamylase, right? Now this transcarbamylase, it will form the carbamoyl aspartate. Now this carbamoyl aspartate will leave out one water molecule by the enzyme hydrotase. Now this will finally form the you can say hydrorotate. From there here hydrorotate again by virtue of the another enzyme, this is known as the hydrorotate dehydrogenase, you are going to get the orotate, right? This orotate, here the Prpp will get involved, the Prpp, right? Now this Prpp actually it will get involved along with the enzyme orotate phosphoribosyl transferase to form the OMP. This is the OMP will form. So it is now slowly taking the structure of the pyrimidine, right? Then OMP decarboxylase will come, and finally you are going to get the uridine monophosphate. It is the parent compound. Just like IMP, it is the UDP for the pyrimidine, right? So this UMP get converted into UDP to UTP, right? Now this UTP will finally get converted into CTP. So the UTP and CTP, these are, you can see relate, these are the nucleotides of the RNA, right? So this is a cycle I am showing you. Here the see this cytosine, it get converted into uracil, and this uracil also get associated with the uracil orotate. It get converted, it get combined with the PRPP to get converted into OMP, UMP. And regarding this, we have already studied that in the previous slide, right? And the next one is the thymine combination with the deoxyribose 1-phosphate to get the thymidine. So this is the overall reactions which takes place. So here you are getting the uracil cytosine, and here you are getting the uracil cytosine, and finally the last product, right? So if you liked the video, just press the like icon. You can share this video with your classmates and colleagues, and for more information from this channel you can press the bell icon. So thank you once again everyone for your patient hearing.